Good afternoon, ladies and gentlemen. On behalf of Business Metropole Ruhr, allow me to extend a warm welcome to you. And welcome to Invest Ruhr Biotech Drug Discovery. Let me briefly introduce myself. My name is Markus Querfurt. I'm supervising innovation networks at BMR, and I'm delighted to be here today to guide you through the event. BMR is the regional economic development agency for the Ruhr metropolitan area, and we are operating for 53 towns and cities and over 5.1 million inhabitants. One of our goals is to promote and connect this high-performing and innovative region, both regionally and internationally. International cooperation is crucial on the path to developing new cancer therapeutics. So our objective today is to connect international cutting-edge life science biotechnology hubs and startups under the topic of drug discovery for new approaches in cancer therapy. We are also happy to offer you the possibility to get in touch with all participants of the event in order to broaden your network, discussing your ideas or forging new alliances in research or business. Today, we have some top representatives of their organizations and businesses as speaker on our event. I'd like to welcome Dr. Mike Bührmann, Coordinator of Drug Discovery Hub Dortmund, Dr. Bert Klebel, CEO of the Lead Discovery Center Dortmund, both representing the biotech hub of Metropole Ruhr, Mrs. Sarah Bruce White, Event and Mission Manager of MedCity London, Dr. George Foote, co-founder of Sixfold Bioscience in London, Dr. Raimund Fickert, Project Director and Head of Communication and Business Development of the Barcelona Biomedical Research Park, and Dr. Laura Folland, co-founder of Bioinformatics Solutions in Barcelona. Be welcome and thank you so much for being here today. Before we go on, I'll hand over for a short welcome message to the CEO of Business Metropole Ruhr, Mr. Markus Schlüter. Due to commitments, Mr. Schlüter is unable to welcome you in person today, but he did not want to miss the opportunity to send you a welcome video message we recorded prior to this event.
Ladies and gentlemen, on behalf of Business Metropole Ruhr, I am very glad to welcome you to our digital event. Business Metropole Ruhr is the economic development agency of the Ruhr metropolis. We develop and market the region. We bring together innovative startups from target markets with actors from the whole Ruhr area. Against the background of the COVID-19 pandemic, the expansion of international cooperation is becoming even more important. With events like this, we want to broaden and to deepen the international exchange of knowledge, and we want to create new opportunities for innovation in the lead markets of the Ruhr metropolis. BMR is eager to welcome and bring together for the first time 3 biotech players of Europe, with Barcelona, Dortmund and London in our talks. The Biomedical Research Park Barcelona is the leading Southern Europe biomedicine hub, and as well as MedCity London is London's heavyweight. We are looking forward to hear from them and their startups about innovative solutions in drug discovery for cancer treatment. Biotech in the Ruhr metropolis is a growing sector in one of our largest lead market, the health industry. The corporate landscape is broad-based with over 6,500 players and about 350,000 employees. The portfolio ranges from biomedicine, medical technology, healthcare IT, and healthcare logistics. The Ruhr metropolis is an attractive and highly productive trade and service location with an annual turnover of over 20 billion euros. In this ecosystem, there are 4 operating biotech technology centers and the Drug Discovery Hub Dortmund with strong ties to university research, Leibniz and Max Planck Institutes. The topic of biotech in times of BioNTech is currently in constant focus of the public, providing never-seen-before solutions in combat of the COVID-19 pandemic. In the Ruhr metropolis, ambitious new approaches in biomedical cancer treatment are being developed and implemented in this innovative field, and we are willing to grow. Today, we bring together biotech actors from Barcelona, London, and the Ruhr metropolis so that they can learn from each other and explore more cooperation. This journey is only possible with our partners. I would like to thank Bioindustry, Centrum for Entrepreneurship with its excellent startup center, Science Entrepreneur Club London, Drug Discovery Hub Dortmund, MedCity London, and Biomedical Research Park Barcelona for their collaboration. Thank you for joining us today. I hope to see you soon in Barcelona, in London, or in the Ruhr area, and I give the spotlight back to our moderation.
Thank you. Mr. Schlüter mentioned them before. I would like also to give a big acknowledgement to our co-organizers of this event, namely Dr. Katrin Dirkes. Katrin is Managing Director at Bioindustry EV Association, and she's coordinating biotech activities in the Ruhr region. Bernd Rathmar. Bernd is Innovation and Transfer Manager for Biotech and Chem at CET, Center for Entrepreneurship at TU Dortmund University. Last not least, Tobias Wingbermühle. Tobias is founder of the Science Entrepreneur Club London, promoting the life science startup community London, Cambridge and Oxford. He's also co-founder of Cluster Market. This event would not have been possible without your support. Thank you very much. Katrin, Bernd and Tobias are having a look at queries coming up and will bundle them for a short question and answer break between the talks. You have also the opportunity to meet them in our networking session after the talks and are willing to give you information about their representative startup ecosystems. That said, you are invited to ask questions to our speakers at any time through the chat window on the event page on our Matchmaker Ruhr platform. If you haven't logged on yet, you can do so using the link we sent you by email before this event. It's easy to register. You can use your LinkedIn or Google account for it. It's free, of course, and it opens you the opportunity to get in touch with other participants in the one-on-one networking session after the talks. Enough said, I'm looking forward getting new insights into the stunning work and research in drug discovery for cancer treatment from our speakers across Europe. I wish everyone here watching an inspiring and engaging afternoon. Let me now introduce you to our first speaker, Dr. Mike Bührmann of the Drug Discovery Hub Dortmund. He is with us today because scheduled Professor Dr. Daniel Rao must attend as an expert in an urgent municipal meeting regarding COVID-19 response, which makes his participation in today's event impossible. We are sorry for the inconvenience. Dr. Mike Bührmann is coordinator of the Drug Discovery Hub Dortmund, working closely together with Professor Dr. Daniel Rao. The science cluster of DDHD employs organic synthesis, biochemical and cellular compound screening, structural biology, structure-based design, as well as target identification for the development of inhibitors and functional probes to perturb proteins of interest. A strong focus in the DDHD is on personalized medicine for the treatment of cancer, especially to overcome acquired drug resistance in non-small cell lung cancer and gastrointestinal stromal tumors. Mike gained his PhD in medicinal chemistry and chemical biology at TU University of Dortmund, and his focus is on the function and biological processes of protein kinase regarding drug development. Mike, the spotlight is on you now.
Yeah, thank you very much, Marcus, for the nice introduction. And I'd also like to welcome all attendees to the speaker panel of this event. I'm happy to take this opportunity to speak on behalf of Professor Rau, who can't attend today, as Marcus mentioned, and to present to you the Drug Discovery Hub Dortmund, or DDHD in short, and to also show you how it ties into the topic of academic drug research. And hopefully you can also answer the question if this concept can actually work. Please let me start with this figure. It is depicting the increase of value generation along the value chain of medicinal chemistry that might be familiar to most of you. We start out at the basic level of basic academic research, where it's all about generating target ID or hit compounds, which are of high scientific value, but not exactly of a high commercialization potential. To this point, we get if we further improve in laborious optimization procedures those hits to so-called lead structures. These are actually interesting for pharma or biotech companies and have very good commercialization potential. And the ultimate goal is, of course, to get a preclinical candidate or even a compound that can be marketed in the end. This process you can also compare with the refinement of diamonds, so to speak, starting from charcoal to a rough diamond to actual brilliance. Now, what's important along this line and why it's often failing when we think about academia is that there's a critical point that needs to be reached, and this is the generation of the lead structure. And here's one concept we came up with why that is. There's a so-called innovation gap between academia, which is very strong at the early phase of medicinal drug development, and industry, which is actually very strong in the later phases of these processes. And this gap needs to be improved. We think there should be mechanisms, efficient mechanisms to facilitate the transfer of innovation to actual application. That means from academia to industry. To further illustrate that, you can think of the so-called valley of death that's between academia and industry. Both institutions have their strengths, their expertise they're good in, but not only in Germany as depicted here. I think it's in many places there's a profound structural problem that leads to lost opportunities. And what one would look for are efficient mechanisms that are actually bridging this valley of death. And these are what we call translators. And how such a concept can actually look like, I'd like to exemplify with the situation in Dortmund. Actually, around the university campus here in Dortmund, we have a lot of different institutions and small companies settled. And this network, as is depicted here by all these lines, are actually real collaborations that are taking part, have been taking part in the past, and are taking part right now. The different partners here in Dortmund, we have on the one hand the MPI, that is very strong when it comes to compound screening, professional library handling, and also structural biology. There's the Leibniz Institute IFADO, which is very strong in mouse studies and tox experiments. Another Leibniz Institute is the ESAS, which is strong in the cardiotox and biomechanical analytics. The Lead Discovery Center here in Dortmund has strong expertise in medicinal chemistry and the whole area of pharmacology. Taros is a CEO for organic synthesis able to also conduct big-scale synthesis of compounds when it's needed. And of course, we have the Biomedical Centrum here in Dortmund, which is sort of the connector when it comes to biomedical questions. And we have the TU Dortmund University right in the middle of it. And in the past, this collaboration network has essentially been used on projects that were funded by the state of North Rhine-Westphalia here in Dortmund. And here's one example of a project that was revolving around the protein kinase EGFR, where Taos was actually capable to produce large amounts of a precursor for further synthesis, establishing an SAR, generate such a compound right here. We forwarded to mouse studies that were conducted at the IFADO, and the LDC was actually capable of characterizing the tissue samples derived from these studies and transferring this knowledge back into further optimization cycles of the compound. And this was the ideal situation to successfully apply. Of course, there's also preventers. I will come to this later, another partner. This complete portfolio of FFC was the ideal situation to successfully apply for the DDHD, the Drug Discovery at Dortmund, which is an EFRE-funded project where we have these 8 mentioned institutions that are sort of the pillars of the DDHD right here, and with the goal to develop an actual infrastructure for drug discovery. The DDHD was funded in April 2018, and what makes it unique is that there is this whole infrastructure. This is one part of the funding where we acquired lots of instruments facilitating our research. Another big part are proof of concept studies that are funded. And of course, another part is education and entrepreneurship. And here's one more figure to illustrate how this whole collaboration network works, and to remind you that this research is not a linear process. It's more kind of a zigzag along this value chain of medicinal chemistry. It's a ping-pong game. Results that are generated by the academic partners are passed on to other partners in industry or other academic partners. They generate results and they're fed back into the infrastructure available at other institutes and so on. Of course, each partner has their own expertise and are strong in certain things. And this infrastructure helps us to really make efficient use of this. And these are, once again, the main goals of the DHD: to strengthen the network that was already existent in Dortmund and make a real infrastructure out of it and optimize it. And to successfully test and validate this infrastructure, there are so-called demonstrator projects within this DDHD package. And one of which I will show you as sort of a case study to exemplify how such interaction can work. And here we come to the topic of targeted inhibition of the protein kinase AKT. It consists of 2 domains. One is the kinase domain and the pH domain associated with it. And one way to target or inhibit this kinase is the traditional approach with ATP competitive inhibitors. One of this is capivasertib. And one unique thing about this kinase is that it also has an allosteric binding site that leads to association of both domains and thereby inhibit the kinase. And one example for this is the allosteric interdomain inhibitor MK2206. The research on our group with Professor Rao led to the development of the drug, or the compound berosertib, which combines the allosteric mode of action with the irreversible inhibition of the kinase, addressing a cysteine residue within this interdomain region by adding a microceptor moiety to this molecule. So upon an initial reversible binding event, the compound is capable to form an irreversible bond with the kinase and thereby effectively inhibiting the enzyme. And borosertib is an example of a first-in-class covalent allosteric AKT inhibitor, as we call them. We were able to generate a whole package of promising data with this compound. It is very potent in cells. We could show the successful covalent modification of the target protein. We have an excellent selectivity profile across the kinase subfamily. In fact, we are only significantly addressing the AKT isoforms. And we have a very robust and reliable crystallization system, which leads to, with which we can generate complex crystal structures that gave us useful information towards the optimization of those compounds. We also have data on bioavailability of the compound and initial data on tumor growth inhibition. But there's one major limitation of this compound. Once again, this illustrates the excellent selectivity and the beneficial tox profile of this compound. But there are liabilities when it comes to parameters like solubility, clearance, or the efflux parameters, or efflux from cells, that is. So there's an urgent need for compound optimization, and this is where academic research is pushing the boundaries of its limits. And this is why we made this covalent allosteric AKT inhibitor project, one of the demonstrator projects called DDHD. And together with our partners at Taros, IFADO, and the LDC, we were able to optimize the parental compound borosertib in multiple optimization cycles and generate a number of frontrunners that show improved features and actually overcoming the mentioned efflux liability, and furthermore shown an improved oral bioavailability. And they're currently, as we speak, forwarded to in vivo pharmacokinetic studies, and also with the aim to subject them to efficacy studies in mice.
Mike, you have 1 minute left.
Yeah, I will get to my final slide now. So to answer the question, can academic drug discovery actually work? I think with the example of the DDHD here in Dortmund, we can answer it with a yes. What it needs for this is the highly complementarity research structures that are located in close proximity within one region, making it able to converge all the expertises to drug discovery and development. And knowing that there are also additional potential partners in the state of North Rhine-Westphalia, when you think of including also the clinics, there's a chance for strategically building an even more bigger and proficient network. So the DDHD is a prime example for an innovative way to shorten and accelerate the path from basic research to drug development, and serve as a successful concept of what I first mentioned, that there's a possibility for transferring innovation to application, the basic research from academia to industry. And with this, I'm at the end of my talk, and I want to say thank you to all of you who listened, and I'm happy to take any questions.
Okay, thank you very much, Mike, for your inspiring words. I would like to ask Bernd, are there any questions from the audience to answer for Mike?
Thanks. So far, I did not gather any questions, but maybe Mike, hi. I can ask you some questions. First of all, thanks for the really nice overview. What is happening in Dortmund since a decade? I think that's pretty impressive. But of course, from my point of view, so I'm also a scientist, a bit ago, but now I'm really want to, let's say, give more attention on the topic of entrepreneurship. You mentioned it, education. So from all these facilitators and all the things which will be in the future, What do you think, or what are right now the, let's say, concepts to really foster more of this? Because of course we have the infrastructure in Dortmund, but we need the people to use it and give them also, let's say, the space to work on all this stuff, because commercialization is just let's say linked to people, which are really boost up these commercialization. I know you mentioned Provendus, but in the end it needs people who like to start maybe their own company, but based on basic research, which has a really high potential to commercialization. So any guess, or from the coordinator point of view, what's really going on this topic?
Entrepreneurship and how to say that, to consolidate the projects we're working on in the DHD, there's of course always the wish to like start a startup company or spin-off from those projects. It really depends on the results one achieves, right? And you know, of course, here in Dortmund, not so long ago, we had the founding of the company Per River, which actually was deriving from one of these academic research projects. And if I'm not mistaken, there are also activities to create the space for those emerging companies, I think in the harbor area of Dortmund, for example. And yeah, the whole startup scene is kind of booming. So Dortmund can't be the only thing or only example here.
Thanks. So there's right now it's popping up one question in the chat box. So from Nicolas, are you using machine learning algorithms so far for drug discovery, or any new, let's say, supporters or strategic partners in your network?
Machine learning, not exactly. Of course, computational chemistry is playing a growing role also in what we are doing. We also have potential partners on hand who are capable of covering this area of machine learning. But these projects we are covering in the framework of the DHD, they are not making use of this right now.
Thank you very much for this question and answer round. We come now to our second speaker from the biotech cluster Dortmund, Dr. Bert Klebel. Dr. Bert Klebel has gathered more than 20 years of professional experience in the life science industry. He has worked in different positions in drug discovery and early development, most recently as Senior Director, Discovery Biology and Head of Biology as GPC Biotech. Before that, he was Axiom's Vice President Research, responsible for the discovery and development of the company's portfolio of kinase inhibitors for various therapeutic indications. He earned his PhD in biochemistry at the University of Constance, Germany, and did postdoctoral work at the NRC Biotechnology Research Park at Montreal, Canada. During his research, he has published more than 55 articles in peer-reviewed journals and co-invented more than 25 patents and patent applications. He's now director of the Lead Discovery Center Dortmund, and we are happy to have you here. So I'll give the word to Bert.
Thank you very much for the kind introduction. So then I shoot off with the presentation, and basically I'm talking now about the Lead Discovery Center, an organization which is actually and truly bridging academic research into commercial application. We consider ourselves to be a translational research center, and basically we have been started out by the German Max Planck in 2008 with the purpose of primarily serving the drug discovery incubation first. So initially there was a strong bias with Max Planck projects, but later on we had the chance to open our premises also to other partners through joint grant application, which was outlined by Mike just a second before. But we also continued the incubation of our Max Planck projects in an infrastructure which you actually see over here in a nutshell, which is basically covering these different disciplines like biology, which is more or less a department for profiling compounds in different various biological and phenotypical assays. It's medicinal chemistry. That's the department where we create all the novel compounds depending on the data which we generate. We have the screening department, which we share with the local Max Planck and also a bit with the TU Dortmund. Pharmacology department is doing what Mike just showed, a lot of the bioanalytics, in vitro and in vivo, the ATME assays. And then we have a small site in Munich for small therapeutic antibodies. So that's the setup. And basically the whole organization is built on a matrix structure, is running X number of projects. And as I said, it's linking academic research and commercialization. Due to the link to commercialization and the successes in commercialization, we also managed to set up a small fund-like structure at the end of 2019 to further support early-stage drug discovery projects, which really are knocking at the door to translation. So our main motivation is really to increase the rate of cures, be it Alzheimer's, be it cancers, or be it even novel pandemics. And we completely acknowledge that basic research is out there to identify the causes of a disease. And this is often taking place through a year-long research and basic research, and then identifying a molecular mechanism for a certain pathology. But it needs much more to happen than just a miracle, which these guys here point out, to translate that finding into a drug on the market for the patients. And it's also much more than a gamble. What we observe in the industry, for example, is that you have to go through around 10,000 new compounds. If we talk about small molecule drug discovery, medicinal chemistry, natural products, we have to go through the iteration of 10,000s of compounds to identify one which eventually will make it to the market. And of course, this has immense costs. It's including all the attrition. A new product on the market for the patient will cost more than 2 billion euros from scratch all the way to its market launch. And we believe that's, first of all, very inefficient to come to synthesize 10,000 compounds to come up with a new drug. And secondly, this is way too expensive for all the health systems to be actually maintained like this. So what's our measure against it? We try to translate basic research where there is really plenty, particularly if you talk about Germany. Germany is spending enormous amounts on an annual basis in biomedical research. So there's plenty of excellent basic research, which we try to team up with our department, our organization here. And what we do is not much more than providing high quality drug discovery services. And in order to incubate this research over a number of years, and then eventually to find a partner for subsequent licensing or for a spin-out company. You've already seen this kind of image. What we really think is we are an organization to bridge this valley of death. So linking basic research to applied research, to pharmaceutical research, to commercialization. I will have, in the course of this talk, I will have a short example on a project of inhibitors of the mitochondrial translation to show you how this really works. But first of all, a bit more information on the organization. I already introduced you a bit to the structure. And although we have like approximately 100 people working in these different departments, it's still a semi-virtual organization. So we consider the LDC, like to provide you with an image or an impression, as a spider in the academic web. So basically, LDC sits there and is offering this kind of services and then collaborating with all sorts of different academic institutions. And of course, preferentially with the ones which are in geographical proximity, like the Ruhr area or the Rhine-Ruhr area. I already mentioned that we are around for 13 years now, being initiated by the German Max Planck and having received significant financing, project-based financing, also from Max Planck. So this doesn't mean that we run grant applications. It's really a collaboration and an exclusive, sort of a preferred partnership with the Max Planck Society to mature and to valorize their project proposals and their ideas. And based on hooked on the success here, we are also able to raise this little technology transfer fund here. But what's also very worthwhile to mention is that we actually created a large, broad academic network. I talked about the spider web and entertain a large number of different collaborations with different universities. Basically, with regard to the profile of our employees, most of them do have prior expertise from pharma and biotech. We entertain around 20 active drug discovery projects at any time, which are completely complementary to the industry. So usually we pick those projects where the industry thinks they're too risky. What's also unique is that we cover a broad range of indications, means that we are indication agnostic. I mentioned the two sites, one, the major site over here in Dortmund, then a small site down in Munich. And what's also unique, I guess, is that we try to partner with industry or investors as early as possible, being supported by a large portfolio. I think that's a quite natural move. And now coming to this example, to the inhibitors of mitochondrial transcription, I would say that this is really a prototypical project at the LDC. It started like more than 10 years ago when we teamed up with the Max Planck director who was back then at the Max Planck Institute for Biology of Aging in Cologne. He now moved on to the Karolinska in Stockholm, Sweden, and we still collaborate with him and his partner, Claes Gustafsson, who is actually a professor at the University of Gothenburg in Sweden. So they have been both instrumental and also essential in starting and supporting that project. And we really started out in the early days just knowing the target. And what's the target? The target is the mitochondria or oxidative phosphorylation. So what we really try to do here is to block the function of the mitochondria. Why do we do this? First of all, there's been described the tumor dependence on oxidative phosphorylation on the one hand. Secondly, quiescent cancer cells, they do show a high requirement of mitochondrial respiration. And as a third point, there is in the abnormal tumor vascularization, there is a high energy demand. And by blocking this, by inhibiting the mitochondria, we and our partners thought it's a good strategy. The rest of the industry perceived the idea more or less like this. So they really thought that's a funny idea, messing around with mitochondria. Most likely means that we will not just kill the tumor, but also the patient. Our partners, but they were actually on a mission. So this is really very clear. They were always convinced that you can block this enzyme, the mitochondrial RNA polymerase, and they provided ample evidence from their basic research that this truly is the case. In between, we confirmed this also in the peer-reviewed literature. Just recently, late last year, we published an article in Nature, a very well-known scientific journal, where we demonstrated that, in fact, you can actually inhibit the human mitochondrial DNA transcription. How did we do this? We used the mRNA polymerase as a target, screened around 430,000 compounds, and moved these compounds through a profiling cascade, various different orthogonal assays, secondary assays, counter assays, and selected finally 55 validated hits as starting points for our medicinal chemistry optimization cycles. So it means, first of all, to improve affinity, potency of these compounds, but also to improve the pharmacological parameters. And this is what you actually do in a typical profiling cycle like this, combined with medchem cycles. So these are iterative cycles. Each one lasts around 2 to 4 weeks. And you actually create new compounds, and then you test the compounds in a profiling cascade like this. So we were able actually to move these initial hit series and increase potency by and large. You can actually see that we have, from some of these series, we now have sub-nanomolar IC50s for some of the inhibitors, which is pretty potent. The more you optimize these compounds, the sooner you get to the in vivo experiments. And one of these examples is, of course, here. That's one of the representative compounds which we tested in cells. And you see actually prominent inhibition of cancer cells, or same is actually true on spheroids. And here in the cancer cell panel, you see that 1/3 of all different tumor cell lines are being potently inhibited, shown by that bar, by the bars here, which pointing downwards. We also worked on the structural resolution, means that we could characterize the binding of that inhibitor towards the mitochondrial RNA polymerase. So it's a true binder of the RNA polymerase of the mitochondria, an allosteric inhibitor. So this has been resolved. We moved the compound into an animal model, typical xenograft model, solid tumor model, which you can actually see here in the mouse. This is the size of the tumor over here, the black line. If you add the compound, you'll see that the tumor is no more growing, as evidenced here. An important control is that basically the body weight doesn't change. We treated those animals for around 4 weeks with the compounds, and we couldn't see a body weight change, means this was also well tolerated.
Sorry, Bert, time is running up. You have 1 minute left. Thank you.
Okay. And just like this one model, which I showed now here, basically proof of concept is a typical situation where we start commercialization. We commercialized a number of projects throughout the years, as shown here. Typically at the proof of concept stage, these are the most licenses which we stroke, or we started spinouts. The one was mentioned earlier by Mike. It's the Pearl River Bio spinout here in Dortmund. At this point, I just want to thank everyone who was actually collaborating with us over the years. These are the organizations which supported us, the spider web here of the network of collaboration partners, and most importantly, our team. And thanks for your attention.
Thank you very much, Bert, for the insights of Lead Discovery Center Dortmund. I'll ask again, Bernd, do we have any questions to to Dr. Klebel right now?
I don't think so.
Okay.
Not in the chat box, but
may I ask a question?
Yeah, of course.
Laura Forlang. Very interesting presentation, Dr. Klebel. I have a question regarding the assays for toxicity. So this is passed on the collaborations and the different organizations that you find synergies with in a later stages, or do you do the toxicity testing or the off-panel off-target testing in your organization? So how do you manage this stage of compound testing?
Thanks for the question and thanks for the compliments. So yeah, we do in vitro tox testing, and also so we do, of course, in silico tox testing using well-known programs like Derek, for example. We do that in-house. The moment it comes to in vivo tolerability testing, we typically do this with contract research organizations, or sometimes also with our academic partners, if they can basically execute these kind of experiments with regard to oral gavaging or all the different application routes. So it's a case-by-case consideration, but we also have a good network of CROs, which we usually use for the standard routine and toxicity testing and tolerability testings. Particularly here in Dortmund, to mention, we also do it a lot with the IFADO, for example.
Thank you very much, Laura, Bert, Bernd. I think we should go and move on to our partners from London right now. Our next speaker is Sarah Bruce White. Sarah works as MedCity's events and mission manager, overseeing the varied exciting national and international events program of MedCity. Sarah has many years of experience in the events industry, following the last 9 years working at innovative medtech SMEs in the southeast of London, bringing new devices to market and launching them internationally. Sarah has a deep understanding of the challenges faced in this area. Today, we are happy to learn from Sarah about the MedCity Life Science Hub activities in London, Cambridge, and Oxford. Sarah, thank you for being here today. The spotlight is on you.
Okay, well, thank you very, very much, Marcus, for that introduction, and hopefully everybody can now see my screen as well. So great to be here today, and thank you very much to everyone for within BMR for allowing me to present. My name's Sarah Bruce White, as Marcus said. I'm going to tell you a little bit more about MedCity and some of our programs, and also a little bit about London's strengths as a life sciences ecosystem, but also highlight why collaboration is so important. And just to also give you an idea of some of the collaborations in cancer drug discovery, enabling those discoveries to get from the bench to the bedside. Again, if anyone wants to follow up, and because we're talking about collaboration, I'll send the contact details around later. So international cooperation and collaboration is also essential. So whilst I'm probably the least technical presenter on the panel, many of my colleagues have very, very strong research backgrounds, and MedCity priorities are to provide a single front door and concierge service for industry and investors looking for partners, infrastructure, and expertise. So we need to connect those partners to help create powerful scientific networks to tackle the big healthcare challenges of our time and drive growth and investment in the sector for the health and wealth of the nation and internationally. So we facilitate and support collaboration across all parts of the sector to make sure those innovations and commercial products, from cancer drug discovery to all sorts of modalities, do get to market. Ensure we help foster an environment that supports and encourages entrepreneurialism and raises the global awareness of the region's rich life sciences ecosystem. So, with the limited time we have today, I'm not going to be able to discuss everything, but we enable collaboration through the ecosystem through many programs. And some of our programs and services help companies find that all-important investment in space academic collaboration. One that I specifically wanted to mention there. In fact, I will just look at this slide briefly and then talk about some of our programs. So collaborating, locate, growing, investing, they all come together, and having those collaborations between research and industry is incredibly important. So some of our networks, MedCity runs a Collaborate to Innovate program, which the next speaker from Sixfold has been involved with, and that ensures companies working within advanced therapies partner with academic researchers. We also run an advanced therapy network. We have a MedCity investment hub, and we work with the King's College London, UCL, Imperial College, Queen Mary, to make sure that we bring those partnerships together. And we work across all modalities of life sciences, from drug discovery to data and AI. And collaboration and partnership, as I've said, can only really happen as long as we have these network opportunities, a little bit like today's event. And whilst we're all very much based in different locations, within a certain hub, location and innovation hubs can form an important part of that collaboration. I've heard somebody recently say that there can be no life sciences cluster without the science. So proximity to academic capacity and academic researchers is very important, both due to the ideas and the scientific entrepreneurs generating those ideas, and then the teams that can work within them as well. So my next slide has some common characteristics of a cluster of creativity. So London has all the ingredients, as do Dortmund and Barcelona, for the world's leading health and life sciences clusters. So looking at investment, knowledge centres, entrepreneurs, networks, so all making this sort of beautiful pie chart of open innovation. So I'm mainly talking about London, but MedCity work across the Greater South East, so that includes the Golden Triangle of Oxford and Cambridge as well. So being in that sort of triangle means that we come together to be one of the strongest biosciences clusters in Europe. We have 19 of the top 20 pharmaceutical companies. So world-leading financial centre, strong biotech investment, and again, the collaborations there are very important. The UK R&D spend was 37.1 billion in 2008. The academic base, so there's 4 of the top 10 universities, and then world-class research institutes such as the Francis Crick Institute, Harwell campus in Oxford, and the Sanger Institute in Cambridge. So one of the unique things, so and I should highlight here as well that MedCity was set up in 2014 by the Mayor of London and the region's academic health science network. So and they're on this slide. So London's unique in one way that across our innovation ecosystem, we have a vast population of over 8 million and a very diverse population. So when that's joined up and utilising the NHS services, it means there's quite unique opportunities for clinical trials, including in cancer drug discovery, and also for the safe and ethical collection of health data, which is something that's received significant investment from Research England recently in the form of the health data research hubs. I don't have time to go through those, but again, if people want to connect with me in MedCity, I can give information on there. And health data has become something that's very, very important. In terms of clinical trials, there's been over 3,200 commercial trials take place over the last 3 years, and 40% of the National Institute of Health Research's portfolios trials in England took place in London and the Greater Southeast. So as we know, collaboration within academic research and internationally and across London is key. So I was just going to provide a very short snapshot of some of the innovation and knowledge centres that I know other hubs have as well to help develop that interface between academia, SMEs, large pharma, so forth. So one of those, an example of that is the Cancer Research City of London Centre, which I will mention in a moment. But research institutes such as the CRIC came together originally because they would house the scientists, the academic research and so forth. And MedCity helps facilitate that collaboration by convening those stakeholders across the ecosystem, and networking through events such as this are incredibly important. So I do invite people to get in touch afterwards. So this is an example of White City, which is it's Imperial and where Sixfold, who will speak after me, are based. So I suppose the days of R&D occurring, a million miles away with behind closed doors is probably over. So this is just a snapshot of Novartis opening their White City Institute of Biomedical Research at White City, alongside Imperial's Innovation Hub. There's King's Cross, which is another development, which is the Knowledge Quarter. So GSK have an AI centre here, DeepMind, MSD are planning their research hub there. And then UCL, which University College London, have a data and AI centre, as well as the Francis Crick Institute. In fact, I think there was a quote from David Peacock, who was the MD of MSD. I read that in the Financial Times, that they said they view the UK as a world leader in developing science, but because of a long-term emphasis on building a research and development infrastructure. So, but obviously collaborating internationally is incredibly important. So moving on to talk a little bit more specifically about cancer before I move on to an example of cancer drug discovery. So the Cancer Research UK City of London Centre is a centre of excellence in cancer biotherapeutics and brings together researchers from the Bart Centre, University College London, King's College London, and the Francis Crick Institute. UCL also has obviously links to the UCL Hospitals Foundation in Great Ormond Street, which is a world-leading children's hospital, where one of the challenges, and I know one of my other speakers mentions this, is the tackling the challenge of cancer evolution through the development of biological therapies aimed at harnessing the power of the immune system to control a rapidly evolving tumor entity. I think about 400 scientists work at this centre, again, all working together collaboratively. So during the last few minutes, and I probably whipped through this a little bit too fast, apologies, is the Institute of Cancer Research and the London Cancer Hub. So cancer drug discovery, as we know, requires expertise and resources in academic and pharmaceutical and collaborations. But the ICR is an independent research institute based across 2 London sites, and the London Cancer Hub is a partnership between the Institute of Cancer Research London and the London Borough of Sutton, and they've got a new Centre for Drug Discovery. So for those that don't know, the Institute of Cancer Research discovers more new cancer drugs than any other academic centre in the world, and since 2005 it has discovered 20 drug candidates and taken 10 of them into clinical trials. Successful at earning income from its discoveries, it then invests that income back into new treatments, which again, very important. So, and the ICR has been involved in almost 1/5 of all new cancer drugs licensed in Europe since 2000. Speaking with a spokesperson at the ICR recently, collaboration with industry is key, and they are, apologies, they are certainly looking to bring companies into their hub. So at the Institute of Cancer Research, am I nearly at time, Marcus?
Yeah, you have 1 minute left. Thank you, Sarah.
Okay, so the Institute of Cancer Research has about 800 scientists working across the full spectrum of cancer research, from basic cancer biology and drug discovery to clinical trials. So I've sort of briefly mentioned the ICR is ranked top for research in the UK REF rankings, but it does obviously have a number of drug candidates and so forth that I've mentioned. So, and they've got their new Centre for Drug Discovery, has specifically 280 scientists combining expertise in drug discovery with expertise in evolutionary biology to develop the first anti-evolutionary drug discovery program. So it specifically wants to facilitate further collaboration, stimulate innovation and drive partnership with industry. So they have opportunities for the rental of space there, actively seeking companies working within cancer drug discovery. But many of the other innovation hubs within London are also looking to invite companies to come and collaborate with them. And just to finish off, looking to the future, I know everyone will be, there's lots of information about AI and health data and how, as we look to the future, how the sort of cancer drug discovery and AI and research and health data, how that combines for a very, very positive future all round. So thank you very, very much for giving me the opportunity to present. Hopefully that wasn't too fast in my whistle-stop tour of London and cancer drug discovery. And thank you very, very much for allowing me this opportunity. Very much appreciated.
Thank you very much indeed, Sarah. As time is running up, I would say if there are many questions from the audience to Sarah, please contact Sarah later on our networking event on Matchmaker to answer your questions. And I would say we go straight on with a startup of London, Sixfold Bioscience, and Dr. George Hood. George is the co-founder and managing director of Sixfold Bioscience, a Y Combinator-backed biotech that uses advanced nano-engineering to create safer and more targeted systems for RNA therapies. Their Mergo platform has been designed from the bottom up to unlock treatments for potentially thousands of diseases. Prior to Sixfold, George completed his PhD at Cambridge University, where he implemented novel methods for the visualization of nanostructures. Today, we are very curious to listen about this revolutionary approach George and his team colleagues at Sixfold are developing. So I'll give the word to you, George.
Thank you very much for the kind introduction, Marcus. I think for today, what I wanted to walk you through is exactly that Sixfold, what we've sort of been building at the company and sort of why that's important and how we're looking to grow in the future. So I think if you really, for the importance of this talk, what I suppose the central dogma of molecular biology is the flow of information. It's the flow of information from DNA to RNA to proteins, and ultimately what effect those proteins have in your body. However, if you get a corrupted flow of information, what essentially that leads to is a disease. You get the incorrect protein, you get too much of a protein, or you don't get enough protein. And that sort of leads on to disease. And really I suppose the panacea of, I suppose, most pharmaceutical development is how you can control that flow of information. Either you're tackling the, source through gene therapy, or you're looking to actually target the endpoint, alleviate some of the symptoms or target the protein. And so obviously there's been a lot of interest in RNA, and that's precisely why there's been interest, is because using different approaches, what you can do actually is silence or express these proteins. So with RNAi, what you can do is you can, I suppose, tune or stop the protein production of a faulty protein, or where there's too much of a protein. And then with mRNA, what you can do is introduce a new protein, so that cell has a new function. Obviously, that's been super effective with the coronavirus vaccine. Or you can produce a protein that is no longer being produced in a cell. And so obviously with these two approaches, theoretically, what you can do is treat thousands of diseases once you understand the underlying mechanism. But one of the biggest challenges actually with RNA is not necessarily the biology. What is one of the biggest problems is how you can deliver those strands of RNA into the right cell. And so this is sort of like a real repeating history, I think, of RNA therapies. It's, I suppose, back in mid-2000s, there was such an interest in RNA. There was Nobel Prizes, a lot of obviously interest in terms of the outcomes that we were seeing in cells. But essentially the delivery stopped that first wave of development, and pharma and all of the money sort of investment into those companies pulled out. But what sort of happened from that point is there was a couple of systems that were unlocked. So lipid-based systems, GalNAc-based systems meant that actually we could start to deliver RNA to the liver. And that unlocked huge value in RNA-based companies. And so this is obviously a graph of market cap of Alnylam and Ionis. And the valuations of these companies are skyrocketing, especially with the recent success with coronavirus vaccines. But if you really look at the delivery systems, they're still focused on the liver. And so all of these companies with these huge valuations and attracting lots of investment are suddenly asking, okay, we've been to the liver, what next? And how do we take these amazing therapies to more patients? And that's sort of really what we at Sixfold do, is essentially think about how you can get the right drug to the right cell. And I think really when you look at the development of RNA, and this is also sort of really a generalization of RNA therapeutic development, typically what happens is you have an academic group working on the therapy, understanding the underlying biology of the disease. The IP is then spun out of the academic lab into form a new company. Lots of money gets put into the company to develop the therapy. And ultimately the investors in the company will think, okay, how do we get it to market quickest? And that is the point of which an off-the-shelf delivery system is thought about. And there's obviously success within the liver, but ultimately to other indications, you get failure. And I think for us, what we saw at Sixfold was that the delivery process, or thinking about delivery, is actually coming 2 steps too late. And really what you should do is be considering the delivery a lot earlier on in the development process. And so when we think about ourselves as a company, or what people, I suppose, should be thinking about from a holistic point of view, is not necessarily, are you an RNA therapy company or are you a delivery company? But ultimately what it's about is actually integrating both of those into the development to create a holistic solution. And so when we think about what could an integrated system look like, for us it's about having a system we're able to essentially iterate and engineer and test a variety of different things so that we can come to the desired outcome. And so this is sort of really the basic principle of Mergo, of our system. It allows us to really quickly iterate through different molecules, through different compounds, to make very specific changes. And that allows us to sort of do 2 things really. the first thing is why this is important is because it allows you actually to break away actually from a scientific hypothesis and actually move into this engineering approach where what you can do is you can take multiple different tests. So kind of like what you have in I suppose when you're designing a website, really, you have an A/B test, and what we can do then is each iteration we can test in and out different molecules, different modifications, different structures, and you can test it. And so through this process, what you essentially get is you get a building of that, the properties of the compound until you get to that desired clinical outcome. The second reason why this is really important, and I suppose mostly thinking about oncology now, is because with obviously what we're learning through genetics is that patients and diseases are very individualized and to overcome some of the challenges of some of the biggest diseases such as oncology, what you have to do is start to take a more personalized approach. And so by being able to iterate, what we can do is actually take the learnings from that data gathered in our previous tests and actually build that and start to build a model or a pipeline where we can actually predict then what molecules we should be building from scratch to treat patients. And this is sort of a really interesting solution to treat a lot of different diseases. And I suppose to date, what we've been able to show with this Mergo system is really building towards that desired outcome that we set in place at the start, which is having an effect in cell, and importantly having that effect in mice so that we can transition that learning into patients. Importantly, using that iterative approach and that engineering approach, what we can do is actually take those learnings forward and tune to different cells. So we know now that when we make certain small modifications or large modifications, that actually has an impact in a biological system. So this is, I suppose, a little shorter piece about Sixfold, the company, and how we got to that, the point of which we are now. We, I suppose, uniquely for a biotech, actually founded the company from scratch. We really identified this problem of delivering or engineering a system to treat any cell, and built it from that perspective of first principles. And so we raised our first initial funding through Y Combinator, which is a hugely successful business accelerator based in the US. We raised our seed round through Y Combinator and have used that money really to develop the team and the technology. So we're based in West London. We're based in one of an Imperial owned building. We've got a fully equipped chemistry, biology suite, which allows us to do all of the internal R&D ourselves actually, and only use, I suppose, CROs for most of the animal work. And then obviously we've been able to build through this. And obviously, when you're taking on a huge challenge, you can really attract some of the top talent from across the world to come and work for you. So we've got a team of 17 people with postdocs and PhD experience from all over the globe, from Oxford, Cambridge, the Francis Crick, UCL, Imperial, and what we can do, like I say, is not just have the capabilities to do this, but also the knowledge in-house to iterate and test, and that's been really important for us, and building in that knowledge into the company. But I would say that we obviously don't act as a lone, isolated company. Through a variety of different programs which have been sort of talked about previously, we're actually able to tap into a wider network of knowledge. So whether that's looking at microscopy, thinking about how we can build specific assays, how we can test, how we can design, these are all really important. And by tapping into these networks and support from different funding bodies, what we're actually able to do is really grow quickly as a company and test and iterate through a variety of different systems. And so I suppose Mark is a little bit ahead of time in getting to the question part, but maybe we can leave it open for a little longer on the question side.
Thank you very much, George, for sharing the inspiring work of Sixfold. You were perfect in time. Nothing ahead. So I'll ask Tobias, do we have questions for George from the audience?
Yeah, thanks, George. Thanks, Sarah, for the very interesting presentation. That was very insightful. But maybe the question from Matt is for MedCity, but we can also use it for George. I mean, George, your journey is quite unique, and your founding team is very diverse from different universities in London and Cambridge. You experienced the support of the Y Combinator in the US, so you really got into the science ecosystems in Cambridge, London, the US. What do you think have been the key support tools or initiatives that helped you to grow? Because I remember when you've been with 8 people in this very small room, in the very small lab, and now you grew to a large R&D team and a large lab in the incubator. So what have been the key initiatives that helped you to come to that stage?
Yeah, I think actually one of the key things is actually the starting point, I would say. I think if you're going to like take a company from scratch, you have to get to that first initial, I suppose, like killer experiment pretty quick, or build a team pretty quick. And one of the things we're actually quite fortunate with, which I say would actually be a limiting factor for most areas, is actually access to space and access to talent. And so getting that first initial space was actually, I suppose, the most important thing. So having I suppose, universities such as Imperial running a space which we could move into quickly that was actually fully equipped, well, not fully equipped, but enough for us to get going quickly, was actually fundamentally important for the success of the company. And through that, then you essentially snowball, and you meet more people. You are able to build those networks because people know that you're, I suppose, an established company at that point. Yeah.
And interestingly enough, none of you is from Imperial, right?
No, I mean, some of our team is from Imperial, so we have researchers who've done their PhD at Imperial, but we're not directly affiliated with them.
Yeah. But I think also Sarah mentioned that example from the White City campus from Imperial College. I think that's something that's Imperial College has done very well. They opened up to any kind of startup. It didn't have to be a spin-out from Imperial, or it didn't have to be a PhD or alumni. They opened up that space for startups, spin-outs from Cambridge University to larger corporates like Novartis. So that's how this ecosystem got together, and that's I guess quite helpful for you to grow, if I'm not mistaken.
Yeah, having people like next door, and like we access the facilities. So we had quite good timing actually, because I suppose when we moved in, the Imperial Chemistry department literally opened maybe a couple of months after we moved in. And also they have like a brand new suite of like every sort of type of machine you could want to use. And so having that next door is actually super helpful, because it means you don't need to like travel anywhere. You just like literally walk. I think there's actually a tunnel. There's a tunnel. You can just walk between the two buildings and transport your chemicals through. So that's like, yeah, unbelievably good. And there's a new, obviously, bioengineering department opening up as well. So
yeah, there's a secret tunnel to the NMR facility, right?
yeah, there is.
Hey, thank you, George. Thank you, Tobias. We have to move on to our next speakers. Our next two speakers are from Barcelona, an incredible place that was my hometown also for some years. So let me introduce you to Dr. Raimund Fickert. Dr. Raimund Fickert is managing the area of communications at the Biomedical Research Park of Barcelona since March 2003. He is also responsible for the business development and directing projects in Barcelona's Biomedical Hub. Raimund holds a PhD in biology from the Institute of Genetics at the University of Cologne and studied journalism at the Institute of German Economy. After his studies, he worked for German pharma industry before he went to Barcelona to support the evolving biotech startup ecosystem with his expertise. Raimund has seen Barcelona's Biomedical Research Park growing up since he is in it from the very first day, making him an unparalleled speaker for his institution. We are happy to have some insights into the PRBB today, and Raimund, the word is yours.
Thank you very much, Markus, for this kind introduction. Thank you to all the other speakers, to the interesting talks that we heard before. I will switch to a presentation, and I welcome all the audience to Barcelona, to the Barcelona Biomedical Research Park. We say that we connect science and diversity in a unique creative space. I think we would all agree that drug development is a very complex procedure, yeah. And I don't know anybody yet who has an efficient way to develop drugs. I think everybody is suffering, and we are trying different things. At least we at the Mediterranean in Barcelona, we could not lose a lot. So starting with the talks of Mike and Bert, they talked about the Valley of Death. Well, we painted our Valley of Death in a different way. We put a conference hall with a layer of water, some palm trees, the beach and the Mediterranean Sea. So this is our Valley of Death. And it's a project that I would want to tell you that is a success story that is unexpected, that is quite unexpected, and that is more focused on academia, on knowledge, than on the development of drugs. The Barcelona Biomedical Research Park is an infrastructure, but it's also a community. It's a community of scientists who, as you see here on this picture, who are talking to each other. That is, it took us 10 years. Here in this picture, you see physicists, you see mathematicians, you see informatics, you see medical doctors. And the big problem in drug development is that you have cell biologists who may not even understand what a lead or a hit is, or how drug development works. So here in our project, we have gotten together lots of a high diversity of different experts who are working and who are talking to each other. Yeah, so that is one of the big advantages. We are public. We are in a science park, and what we do is actually just provide the best possible environment for research of excellence. Yeah, if you want to know a little more, under prbb.org, you can find whatever I cannot tell you now. So the setting is like Hollywood at the beach of Barcelona. Excellent. It's a project of the Catalan government, the University of Pompeu Fabra, and the city of Barcelona. And the park is running now for 15 years. Barcelona has not been known as a hub of biomedical research. So Barcelona was known for different things. So in order to say here we are, we wanted to be at least big. So actually the building is huge. It's not as big as the Francis Crick Institute, but about half of it, 55,000 square meters. Yeah, and it's still one of the largest biomedical hubs, research hubs in Europe. We have several academic research centers with altogether 1,400 people from 45 different countries coming together in this quite spectacular building. Obviously, that was a big question mark. Would Barcelona be able to attract talent from abroad? We saw we could. Once we had created a scientific infrastructure that was comparable to the Max Planck Institutes, to Harvard, to Stanford, we started to compete. And for many years, Barcelona was a very attractive city. Now it's a little different, but it's a vibrant city. And we actually attracted lots of Americans, Germans, Scandinavians, people from UK, who actually found their research profession or laboratory in our building. To give you a little more idea of the size, the annual budget of all the academic research centers is about 120 million euro per year. Yeah. And we have quite some cutting-edge scientific equipment, certain equipment that is not even available in UK and in Germany. You would find here special mass spectrometers or certain type of imaging facilities, for example. One of the main things that we have And that makes all the difference. Yeah, maybe you don't see it too well in the picture. I will put the text a little down. What you see here in the front is actually a hospital. It's called Hospital del Mar. It's a 500-bed public hospital. And behind it, you see the PRBB, the research park, and we are connected with underground and over the ground also corridors. So you can, without any barrier, move from the hospital to the research park. And we have actually medical doctors who are into research of the hospital. They have their research labs located in our research building. The other thing that we have is we decided to open up all the spaces. It's not really an open space laboratory, but the spaces are freely accessible. I'm giving this speech now, this presentation from my home in Barcelona, and I couldn't tell who is actually in my office. Anybody in the building could actually just walk into my office. Yeah, how does it sound to you? It's quite dramatic. Anybody can access any laboratory. Yeah, it's quite complicated to manage because we are not one organization. We are several institutions with different working contracts, but actually the community can freely circulate. That is a big advantage. So the institutes that we have are leading institutes in their field in Spain and beyond. We have the Research Institute of the hospital, yeah, who have their laboratories in our building. We have the Faculty of Health and Life Sciences of the Pompeu Fabra University. That is a young university that was created only in the 80s. And their Faculty of Health and Life Sciences is quite curious. I think it will not exist in Germany because the Faculty for Life Sciences or Biology and Medicine is the same faculty. Yeah. We have a Center for Genomic Regulation that is more oriented in genomics and molecular biology, cell biology. It's considered to be the #2 research center in life sciences in Europe and #5 worldwide. So it's quite successful. We have the Barcelona Institute for Global Health, IES Global. They are more oriented in epidemiology. We have an Institute of Evolutionary Biology. We have a laboratory of the EMBL, the European Molecular Laboratory. They have 5 outstations and 1 is in our building. And we have a PET center, the Institute of Applied Radiopharmacy of Barcelona. Some numbers. The academic community is quite active. They produce 1,200 peer-reviewed articles per year. Of that per year, last year, for example, we had 8 pure Nature, 9 pure Science articles, 3 Lancet, 2 New England Journal of Medicine, and 50 other Nature group journals. That is high impact research. I think most of the researchers are happy and they are generating high impact researchers because they collaborate internally and externally with very successful groups, and they manage to get very good publications. It's also a meeting point for international scientists, at least until last year ago. We celebrate about 300 scientific conferences per year in the building. And some demographic data, we have 170 senior researchers, 250 postdocs, 240 PhD students, 460 research technicians, all those working under one roof in one building. Another success is that we attract quite a large amount of European Research Council funding. There are more than 30 grant holders of ERC grants. And in the last 12 years, we have created about 10 startup companies. It's not a main focus to create startup companies, but if there are researchers or postdocs who want to create startup companies, we give them support. The contents. We do not only research in cancer, but our specialty is really connecting different perspectives. We can say that from bytes and molecules, we research to organisms and populations. So following, you will just see a list of all the different research programs. They are not research groups. Each program has between 5 and 10 research groups. So it goes from cancer, neuroscience, public health, bioinformatics. We have genetics, we have molecular biology, complex systems. We have people working on air pollution, on urban environment, childhood and environment, systems biology. We have systems bioengineering that is led by a researcher who has just arrived from Broad Institute in Boston. He has been involved in the, maybe you have heard that they are generating pigs who will have transplantable organs. Yeah. So
Raimund, 1 minute left. I'm sorry.
Okay.
Thank you.
Okay. And what the researchers also have is excellent infrastructure. We have from animal facilities, bioinformatics facilities, imaging facilities, tissue engineering. And for example, we have a phase 1 clinical trials unit with 12 beds within the research park. In order to create community and trust and for people to collaborate, we have installed like a teaching program that is free of charge, where we offer soft skills like communication, business development, patents, how to create a company, and good scientific practice. We have like between 30 and 40 courses that the residents of our research park can freely use. And if you are interested in the science that is going on, please subscribe to our news channel. It's called Elixir, and you will find out whatever we do, many kinds of cancer projects and other projects. Yeah. Thank you. Marcus, do I have some time? Maybe to mention, if you want to have that presentation, you can just go to bit.ly/PRBB_short. Yeah, you can record the presentation, and yeah, there you have active links to get more detailed information.
We could skip the question and answer section of yours, and I could give you a minute more. But
Okay.
anyway, I think it was brilliant. I think we just have to go on
Yes.
in our time and in our schedule. Thank you, Raimund. If there are any questions to Raimund and the PRBB's work, please connect to Matchmaker Ruhr and make an appointment with Raimund after our event. I would now like to introduce you to our last speaker for today. She's also from Barcelona, from the Barcelona Life Science Ecosystem, Dr. Laura Forlong. Dr. Laura Forlong is head of the Integrative Biomedical Informatics Group of the research program on biomedical informatics and associate professor at the University Pompeu Fabra. She has a PhD in biology from the University of Buenos Aires and an MSc in bioinformatics by University Pompeu Fabra. With more than 10 years of experience in translational bioinformatics, one of her main interests is to translate cutting-edge bioinformatics research into real-world clinical and industrial applications. Her current research is focused in the areas of systems medicine and systems toxicology, text mining, and knowledge management. She has published over 80 peer-reviewed articles and worked in several IMI projects and more. Laura, thank you for being here today, and you have the word.
Thank you very much, Marcus, for the nice presentation. I want to thank you all for being here and also for giving me the opportunity to present our company today. So we'll talk about the potential of unlocking the genomic information for healthcare and pharmaceutical research and development. And so to give you a little bit focus on what are our interests as a company. So we are in an era where the advances in different omics technologies, and in particular those for assessing genomic variants, and our increasing understanding of the effects of genomic variation in health and disease are driving the transition of genomics from the laboratory to the clinical practice. And in fact, genomics is increasingly being adopted in high quality patient care, especially in oncology and also for rare diseases. And its envision and the objective is to extend this practice or this discipline of genomics medicine to other disease areas, and in particular for early detection and risk prediction. So on the other hand, we are also aware that the changing prevalence of chronic diseases and also the emergence of new diseases, as we are all aware of, are giving to the situation that many people are living longer and also with more complex comorbidities. On the other hand, and when talking about therapies, we know that drugs do not work the same way in every person, and this is leading to the emergence of new areas of unmet need for the pharmaceutical companies. But also we are really in a very exciting moment in biology and in medicine, where in the last 20-25 years, there has been an impressive advancement in the mapping of the genetic determinants of human diseases. And you can see here in this nice picture that summarizes the most recent findings. So on the left-hand side, you can see in the case of Mendelian or rare diseases, there has been an impressive advance in the discovery of the genes that are underlying those diseases. And on the other hand, for complex diseases, there has also been advancement in the discovery of thousands of genomic loci. So on the other hand, is to have this knowledge of the associations of genes to diseases give us a powerful tool to pinpoint the mechanisms underlying the diseases, and this is something that has already mentioned in some of the presentations before, and give us the way to design a new and innovative therapies. And in fact, this knowledge is already making a difference in drug discovery. So, and I'm showing here an example of a study performed by AstraZeneca on their project portfolio some years ago, where they showed that those projects that are based on or use the knowledge about the linkage between the targets and the disease have a higher chance of success to later stages in clinical development. But there are still several challenges that need to be addressed in order to make use of all this knowledge that we are accumulating on disease genomics. So one is something that we are all aware of, that is the amount of information, the volume of information we have. So there are more than 500,000 variants associated to diseases, and it's not trivial to identify those variants that are the causative of a particular disease. The other challenge is that of data silos. So there is a lot of information, but it's scattered across different resources that are often disease specific and do not communicate well with each other. And the other challenge that we face is that of interoperability, so that we need that the data is encoded in a way that computers and software can deal with that, can interpret that, and make proper connections between different sources of information, so from more research data set to clinical data sets, for example. And the other problem that we face is that of the publication. So we have a really high speed of generation of publications, and even in topics like disease genomics, there are more than 1,000 papers published each year. So it's impossible to have all these innovations without a use of automated tools like text mining approaches. So in order to address these challenges, we have developed DisGenet Plus. So DisGenet Plus is a knowledge management platform on disease genomics that contains or is based on a comprehensive knowledge database that integrates and standardizes information on disease-associated genes and variants. Is not focused in a particular disease area, but it aims to cover the full spectrum of human diseases and disease symptoms and traits, and also contains information on genes and variants associated to the adverse effects of drugs. And is also an interoperable resource because it's in its design, it follows fair data principles and use community use standards, and in that way can support a variety of applications in genomic medicine and drug research and development. So in order to build the database, we collect information from different resources that are a reference in different disease areas, like databases for rare diseases or resources that specialize in particular data types like genome-wide association studies, and we extract and standardize from these resources information on the disease phenotype and the variant and the gene that are associated according to these databases. And we also extract the information directly from publications using our text mining tools in order to identify relationships between genes and variants and disease phenotypes. So currently the database contains more than 1 million associations between genes and disease, and over 300,000 associations between variants and particular single nucleotide polymorphisms and different diseases and traits. It's important to know that around 70% of the associations are extracted directly from the literature by our text mining tools and are not present in any of the other expert curated resources that we use to develop the database. And in addition to the data, we over the years, and I will explain a little bit about our history, we developed different metrics or scores in order to facilitate the user to navigate, select, and identify the information they need for different applications. And also, we provide different views of the data that can be used depending on the particular application. So for example, if the user is more interested in variant interpretation and variant analysis, it can view the variant disease association. If it's more interested in drug target identification, then the gene disease association would be more useful, or is interested in assessing comorbidities, then the disease association view will be the right choice. So to tell you a little bit about our history, so we are a spin-off company from the Pompeu Fabra University and Hospital del Mar Research Center, as Marcus presented, that are part of the PRBB. So we started our company last year, but we are working on DisCnet. That is a public platform more than 10 years now. So we started, or we released the first version of the public DisCnet tool back in 2010, and over the years we continue the development, adding new information, adding new functionalities, and especially adding new tools for the user to better exploit and support different applications. And this was also possible thanks to our participation in different European and IMI projects over the years. Sorry. So,
dear Laura, you have 1 minute.
Okay.
Thank you.
Finalizing. So just to tell you a little bit more about DisCnet and where we come from. So DisCnet is a recommended interoperability resource from Elixir, that is the European organization devoted to the sustainability of life science data sets, and has engage a large community of users with more than 2,000 citations of publications and thousands of web users. And to show you some examples of this use of DisCnet in the area of oncology is the focus of the meeting today. So here you see some examples of different applications, for instance, for developing tumor therapy based on synthetic lethality approaches, or identification of biomarkers, or investigations of the role of retroviral integrations in tumorigenesis. So other uses of DisCnet is to guide the development of gene sequencing panels, and this is something that is done by different biotech companies that use our data sets. And in addition, our company provides services in other areas such as biomedical text mining, system medicine approaches, and development of data sets for biomedical AI applications. So with that, I'll finalize my presentation. This is a picture of our current team, and I will be happy to take your questions.
Thank you very much, Laura, for sharing highly interesting details of your work. I will ask Katrin, do we have any questions for Laura from the audience?
So far, we do not have any questions. Thank you, Laura, for your talk. But maybe you could just tell us a little bit about how you get access to all the databases. Do you have collaborations with different hospitals? Where you can acquire all the data to the diseases?
Well, the data is acquired from different resources that are not part of our, from different institutions around the world. So, and these are public resources that you can access the data following up their license scheme. So this is the way we access the data.
Thank you, Laura. Thank you, Katrin. I think we will then shortly move on to Matchmaker for the networking session that has been Ruhr Biotech Drug Discovery. A big thank you goes to our speakers for giving us inspiration today from their work. And thank you for watching. We hope you enjoyed the talks today, and be welcome to our networking now on Matchmaker Ruhr. See you and goodbye from Metropole Ruhr. Thank you.